Bacteria are a huge topic that we're just going to touch the basics of the way that bacteria reproduce and the way they exchange genetic material in this chapter. So the genes of bacteria are found in bacterial chromosomes, just like us, but unlike us, they usually only have a single chromosome and it's circular. They may have more than one copy of that identical chromosome, but again, just one of them. Also remember that bacteria are prokaryotic, so that means they don't have a nucleus. Instead, their DNA is concentrated in a place called a nucleoid region. And so these are bacterial cells, and you can see some of these are in the middle of dividing, but you can see that white part is the nucleoid region. It's not wrapped in membrane, but it is where the DNA is located. So here's a bacterial cell, and here's its bacterial chromosome, and this is where the majority of bacterial DNA is found. However, bacteria also have something unique to them, and that's extra pieces of DNA called plasmids. They are also circular, but they're very, very small. They usually only carry a small number of genes on them. And they exist totally independent of the bacterial chromosome. They'll copy on their own, so you can actually have several copies of a plasmid. So you can see in this example, there's 6 plasmids inside of this cell. Plasmids are usually not necessary for survival, so remember that genes that were critical for survival would be found in the bacterial chromosome. But the plasmids often provide some sort of growth or survival advantage. Also, in some cases, plasmids can actually become part of the bacterial chromosome and be permanently integrated into it. So there's 5 big categories of plasmids that tell us a little bit about the kinds of advantages that plasmids can provide. So the first one is pretty obvious, resistance plasmids, or R factors. These are plasmids that carry genes that cause that bacteria to become resistant to antibiotics or other types of chemicals. Some plasmids carry genes that allow the bacteria to digest an unusual food source, like, for example, oil, which most bacteria can't digest. Some plasmids carry genes that allow the bacteria to make chemicals called colicines, that actually kill other bacteria. So this is a way to get rid of the competition near you. Virulence plasmids are plasmids that carry genes that cause a bacteria to become pathogenic. So some bacteria, they are not going to hurt us, right? Many of them don't hurt us. The way that some bacteria hurt us is through their plasmids. There's genes there that perhaps cause diarrhea, for example. And without that plasmid, the bacteria wouldn't cause diarrhea. And then the last one here is a fertility plasmid, or an F factor. And fertility plasmids are going to carry genes that allow bacteria to mate, hence the term fertility plasmid, which is really in bacteria is just going to be an exchange of genetic material. And we'll talk about those in particular a little bit more later on. So reproduction in bacteria is very rapid. For example, in E. coli, they can divide every 20 to 30 minutes. So a single cell can form a colony of bacteria, which is hundreds and hundreds of bacterial cells, in less than a day. Bacterial reproduction occurs by a process called binary fission. So let me just remind you that it's not mitosis, basically. So it's cell division, but it happens without the microtubules, without all these steps that we associate with mitosis. Because it's just making exact copies, unless a mutation occurs, the mother cell is exactly identical to her daughters. So mother to daughter cells, no changes unless a mutation occurs. So this is asexual reproduction, it's not sexual. And then remember that plasmids will be copying separately from the bacterial chromosome and get divided randomly between the 2 daughter cells. So this is binary fission. Good news, you don't have to learn steps of binary fission, but you can see that it copies the DNA and then divides into 2 new daughter cells. And this is, and if you take microbiology, you'll get firsthand experience with making these. These are bacterial colonies that are being grown on a plate. So this actually represents thousands of cells. And because they grew from a single cell, they're all identical. So again, because bacterial reproduction is just making identical daughter cells unless a mutation occurs. So again, genetic diversity in bacteria is primarily only going to happen through new mutations. So a new mutation can alter the bacterial genome and give it traits, new traits. And so this is where some things like resistance come from. So this is one way that you get genetic diversity. But bacteria have another way to get genetic diversity. It's not sex, because that's how we get our genetic diversity. It's something called genetic transfer. This is when genetic material can be transferred from one bacterial cell to another. And again, it's not sex, it's going to be the direct passing of genetic material, but this actually allows bacteria to get new traits faster than mutation alone. And this was really important for scientists to realize, because we were somewhat mystified when we would see things like resistance happen much faster than the mutation rate would predict. Okay, so we know the mutation rate of bacteria, we know how fast they're going to get mutations, but we were seeing that bacteria became resistant much faster than that mutation rate could explain. And this eventually, scientists realized that the reason why they got resistant so much faster was that bacterial cells can pass resistance to each other. So they don't have to get it from a new mutation. It doesn't have to come from a new mutation. So that means it'll happen faster. And that was really important to understanding in the medical field what was happening with antibiotic resistance.